The overall goal of this method is to recover and analyze all the major molecular entities, including polar and semipolar metabolites, lipids, and proteins from a single sample using a simple fractionated methyl tert butyl ether extraction.
Generally, scientists find it difficult to analyze multiple compound classes from a single sample. Using the MTBE extraction that we introduce here, you can extract and analyze multiple compound classes like lipids, metabolites, and proteins from a single sample.
The main advantage of this technique is that it can robustly extract all molecular compound classes from a small amount of single sample aliquot.
This method helps answer fundamental questions in systems biology, since it provides the experimental basis for multi-omics analysis, providing fractions which can be used for analysis by proteomics, lipidomics, and metabolomics.
The following protocol is demonstrated using Arabidopsis leaf tissue. Arabidopsis are small flowering plants in the Brassicaceae family and are related to cabbage. Begin this procedure by pre-cooling the tissue homogenizer tube holders in liquid nitrogen for at least 10 minutes. Remove the samples from liquid nitrogen and place them in the tube holders, and then remove the tube holders from liquid nitrogen. Quickly put the tube holders in the tissue homogenizer and set the homogenizer to grind the biological material into a fine and homogeneous powder. For leaves, use 20 Hz for 1 minute.